I want to welcome you all to our November version of Plant Phenomics Fridays. This is something that's been going on for a few years now, and every month that it's possible for us to find people to give seminars that want to do that, that's in this topic area. And every month that we have the bandwidth to make it happen, we do that. We stream these from Iowa State University. The series has been funded in the past by a number of different grants around here. In the past, the Midwest Big Data Hub was one of the groups that funded Plant Phenomics Fridays, as was the Plant Sciences Institute. Right now, we're funded by HIPS, which is High Intensity Phenotyping Sites, a multi-scale, multimodal sensing and sense-making cyber ecosystem for genomes to fields. This is a Maize project for those of you that are not familiar with genomes to fields, and that is funded by USDA NIFA. We are also funded in our November series by IRA, an AI Institute for Resilient Agriculture that's funded by USDA NIFA, as well as the NSF. And somebody that I always forget to say thank you to, that as long as we're doing this kind of introduction, I want to do a shout out. Darwin Campbell and Rod Fisher are the two people that actually make this seminar series happen. They do the logistics and on the ground stuff that creates a situation where it's easy to tune in, to show up, and if you're on campus at Iowa State, to get a lunch. With that, I'm going to do an introduction for our first speaker today. Alper Adak completed his master's in science at field crop at Akdeniz in the Mediterranean. This is Akdeniz University in Turkey, and I'm sure I pronounced all of that in some iffy way, so I apologize for that. Alper joined the PhD program of the Department of Soil and Crop Science at Texas A&M in 2018, and since then, his PhD research topics have been related to plant breeding and quantitative genetics in maize, more specifically to the integration of high-throughput phenotyping data with high-throughput genotyping data, temporal phenomic prediction, and selection. So with that, I am going to turn that over to Alper, and you can see his seminar title here. Thank you for joining us today, Alper.
Thank you for the nice introduction, Dr. Carlin. Yes, this is my title, slightly long. What I wrote in here is that temporal unoccupied aerial system can inform as much as a genome-wide marker in prediction of unknown genotype in unknown environment. But if I summarize, a little bit more clear, I can say that what I'm going to mention today is phenomic prediction. So this is one of our last publication result, and I'm also going to present this presentation in 3 society meetings. If you are planning to join there, I will be happy to meet in person with you to discuss, of course. And I would like to start with this figure, because this figure first time initiated a phenomic prediction idea in my mind. What we are seeing in here is that, as Dr. Carlin mentioned, this is the genome to field maize hybrids. We planted them in 2017 in Texas environment. So from left to right, what we are seeing is there are 3 different trials. These are drought, irrigated, and late planting, and each contains the same set of hybrids that are around 250 maize hybrids. And what we are seeing in here is that temporal plant height data. X-axis are the days after planting time. These are the flight time as days after planting unit. And the y-axis are showing the temporal plant height scale. But there is a clear pattern we observed in 3 trial consistently, was that the plants with temporally taller plant height resulted in higher yielding hybrids. If you ask how I separate the high yielding and the low yielding hybrids, basically I tag the high yielding hybrids if the hybrids had the higher yield value than the average yield value of each trial. And this is what I have seen. And then I started to talk about that. Okay, temporal data, phenotype data can be a good predictor for us to predict or classify whatever trait we are interested in this case, yield. And also we publish the detail of this research if you would like to, look at the detail. But before going forward, I would like to talk a little bit about our field-based high-throughput phenotyping pipeline. This is not straightforward right now. We are using a bunch of executive function of different external software in our environment. But very briefly, I would like to, we are planning to make this pipeline our package soon, but I just would like to talk about briefly how we are extracting our traits or how we are generating our high-throughput phenotyping data. So basically, we are having two different file type for each flight date, and then we are using the point cloud data to extraction the plant height, and we are using the TIFF file to extract the vegetation indices. In here, what we are seeing is this is basically one of the appearance of the point cloud data. We are constructing DSM, DTM, and the CHM. CHM is what we are using as temporal data, canopy height measurement, also known as a canopy height measurement. This is very typical procedure. Many researchers so far has done these things very successfully. And then what you are seeing here, the plot-based extraction. We are using the polygons or rectangles or shapefile. We are basically aligning these polygons with our plot. Basically, we are using 7 meters long row plot for each pedigree, and then we are calculating our plant height across the top for each drone flight. And then we are doing pretty much the same procedure for vegetation indices, but in this case, we are not using the point cloud data. We are using TIFF file, and we are basically removing the soil, and then again we are aligning our shape file for each row plot. You can see the zooming picture, and we are extracting the vegetation indices. This is again not straightforward procedure, but we are working on it. So this is my first attempt. So what I did in here is that we used 12 flight, and there are 3 trial in here, each containing 100 advanced hybrids. And then they were grown based on the randomized complete block design, experimental design, and then we basically evaluated 800 row plots for each trial. And then the only differences for this trial is planting date. One of them was planted March 21st, one of them was planted April 12th in 2019. And then what we did in here, I extracted various vegetation indices belonging to 12 flight times in here for each trial, and I use this information as predictors to predict flowering time. Flowering times in maize are divided into 2 groups: days to silking and anthesis, since the silking and the tassel are a separate part in the maize. And then I predicted the yield. I'm mostly going to focus on yield today. And also, I would like to talk a little bit about T-blobs or temporal breeding values. We know breeding values. We are calculating breeding values for instance, yield by running the mixed model. But today, I would like to talk about a little bit advanced mixed model, or how I would like to show our vision, how we approach to dissect the temporal trait by using different, slightly, modified mixed model, let's say. This is why we put the T temporal terms in front of breeding values. So yes, this is the nested idea. The thing about that, you have the 30 flight data and you extracted 30 different vegetation indices for 3 trials. That means 30 multiply 30 multiply 3. That means 9,000 column. Would you like to run ANOVA for each one? So this is insane. You can use loop in R, it's fine, but copying and pasting procedure is very painful. I tried, I'm just sharing, this is very painful. So in order to deal with this issue, the idea is simple. We just integrate the flight as a component in mixed model. This is basically the new approach for us how to dissect the temporal trait. That's all. This is for single environment model, and this is multi-environment model. Basically, think about G by E model, G plus E plus genotype by environmental interaction and your spatial range and rows or rep, whatever you have. And then we are just adding the flies in here. I put this term in here because LMER, I'm sure many of you are already using that. LMER tend to treat the flight component here as interaction, but you can force the flight to use as a nesting effect. I mean, I don't want to go into detail, but very briefly, you can treat your pedigrees as nested within flight. I know nested design can be violated slightly in here, since every flight contains the same experiment, which shouldn't be in the nested idea, but every flight, you can see the same environmental condition in every flight. So maybe we can use or we cannot use. So the idea, this is just, we are offering this idea. And then if you use this idea, whether you can treat flight as nesting or interaction, it doesn't matter. So you can use, you can calculate the temporal heritability, which is slightly different than calculation the heritability or repeatability, because traditional heritability or repeatability, because in the traditional version, we are using pedigree variance as nominator, but in this case, we are using the variation of pedigrees occurred across growth for temporal trait. I believe this is biologically more sensible when it comes to calculation, heritability or repeatability. And this is the multiple environment version of calculation of the temporal heritability. And then these are the explained percent variation. When you treat them, when you treat each component here as a random, we can calculate the explained percent variation. This is not surprising that flight explained the highest percent of variation in here. So which is, the blue, pale blue color in here. And then the diamond white diamonds symbol in here are temporal and the value of the temporal heritability. These are locating between 0.5 to 0.75. So I mean, they are temporally heritable. And x-axis, by the way, are showing the vegetation, different vegetation disease. Their calculations are varying, but you can check the ratio of this vegetation. This is very, very simple. And then when we visualize the pedigree nested or pedigree interaction with flight in here, this is what we can see. For instance, let's focus on the CHM, which is the temporal plan height data. I didn't fit, this is the raw data. And then we are calling these things temporal breeding values, like breeding values for each date. X-axis are showing the X date. This is for late planting. This is for optimal planting. I have already introduced in the beginning of my presentation, as you recall. And then what we can dissect in here is that we can dissect the temporal plasticity, because plasticity or reaction has been dissected between environments so far for the complex trait. But what disregarded so far is the plasticity occurred across time point. So G by E by management by F flight, we can add as a different new component to dissect the complex trait based on a time basis. And this is another example. This is the population 1 and population 2. These are two different populations containing totally different hybrids. Population 1, 2017 genotype field project, genotype field hybrids, and population 2, our own advanced hybrids. My PI is Dr. Seth Murray. He advanced this slide. And then LPI late planting, OPI optimal planting drought, OPD optimal planting drought, OPI optimal planting irrigation. So you can see the differences. For instance, canopy height measurement is getting taller fastly when the same hybrid has been grown as with the late planting, because longer day length make the hybrids grow faster. So we can summarize these sentences with this situation with one sentences, but we can visualize this situation by showing that when we use the results of environment pedigree flight component in here. So this is the typical phenomic data, how it looks. So these are the phenomic data. It goes all the way down, and we can, like, there are like hundreds, even thousands the predictors. These are continuous. These are vegetation. This is belonging to multiple time point. Yield, DTA, DTS, PhD, FHD, basically these are the predicted variables. Today we are going to focus on just yield. And then these are hybrids. I'm just showing these things. I'm sure you have already done a bunch of genomic prediction if you're interested in the quantitative genetics, regardless of what kind of trait you are focusing on. But what you use in there is GBS data, essentially zeros and ones. Basically, they are not numeric, but we are pushing the GBS data to be used as numerics. But in here, these are the continuous variable and the real numerics number, where we can compare the prediction ability of the phenomic data with the genomic data. This is what I'm going to show to you at the end of my presentation today. And once we created this phenomic data based on, I showed our pipeline, and this is where prediction accuracy or prediction ability, whatever you would like to assume, this is the phenomic data, as I showed you before. And basically, we are partitioning this thing 70 and 30%. 70% is training data. We are training model in the carrot package by using the different model. And then the 30% phenomic data is a test data. But in here, we are calling this, since we are using the phenomic data, this is what we are going to predict for 30% test data is phenotypic estimated breeding value. I'm sure you are familiar with the GEBV, genotypically estimated breeding values, since we can calculate these things based on GBS data. But in this case, we are proposing the phenotypically estimating breeding value. And then what we are doing is that x-axis are what we predicted, y-axis are the actual value of the test data. So this is the well-known procedure, but the only difference is the phenotypically estimated breeding value. And we are doing this prediction model like 1,000 times to get the real performance of the different model. And this is the difference. The DHAT is delayed planting and the OHAT is optimal planting. I have already shown in the beginning of the my presentation, but let's focus on the yield right now. And then I tried the 5 different model. And first, I would like to call your attention to the linear model. Linear model, as expected, it didn't predict. So these are basically prediction accuracies that were calculated by using the 30% of the test data always, as I showed you before. And then the rich regression predicted the yield like more than 0.5. And these letters are the 2K HSD comparison. So the most important idea in here is that we can predict yield by using phenomic data. So this is a very hopeful result for us, more than 0.5. So if you just focus on just 10% of your data, then this is basically very promising results for 2 different distinct trials. So I have to be a little bit fast. This is the, we published the results in remote sensing. If you're interested, you can look at. Now, this is the 2017 GTF trial. So now I use 2 different phenomic data. One of them derived from the RGB platform, RGB sensor. One of them derived from the multispectral sensors. Multispectral sensors contains 5 bands in it, RGB, red age, and NIR. RGB sensors, as its name suggests, just contains 3 bands in it. And then RGB platform in here was flown 25 meters and the resolution 1 centimeter per pixel, and there are 15 flights in it. This is the first phenomic data. Second phenomic data for the same hybrids, by the way, of course. And then the multispectral platform was flown 120 meters and the resolution is slightly lower, of course. And then there are 12 flights in this second phenomic data. And then this is when I visualize the RGB based phenomic data, this is how we are seeing. Again, very briefly, because of the time constraint, I just would like to show, this is the yield value of each hybrid. There are 280 hybrids in here. So we can separate high yielding and low yielding hybrids based on the temporal value of vegetation indices. For instance, MGVRI. Or the red one is the higher yielding one, always follows the highest pattern. And then the low yielding hybrids also follows like lower. And then you can visually, I'm talking about visually speaking for sure, but you can see the performance of vegetation, the temporal value of vegetation indices. This is the multispectral one. So by the way, there are 504 null predictors in here, and there are 1008 null predictors in multispectral phenomic data. And this is the same situation. And then there are 4 different prediction accuracies in here I calculated. So 70% and 30% I explained. But in this case, irrigated tree in 2017 is a tested environment where I train my model. And then the untested environment is drought trail where I predict the untested genotype in untested environment. This is the most challenging prediction accuracy where we can calculate. So again, there are a bunch of black spots in here, I know, but I just would like to call your attention for this one. GY is the grain yield, and then GY is the grain yield. The bottom one for, we calculate these things by using the multispectral phenomic data, multispectral derived from phenomic data, and the upper one is the RGB-based phenomic data. Just a reminder, RGB has more resolution since it was flow at the altitude of 25 meters. The multispectral one was flow at the altitude of 120 meters. This is very important. I would like to tell something about that. And as you see in here, 0.54. I mean, when I first calculate these things, I thought that, okay, I did something wrong. I mean, this is impossible because this is CV4, untested genotypes in untested environment. I use, by the way, 1000 bootstrap in here. And then we can predict untested genotype in untested environment with the accuracy of 0.57, basically 0.6 for yield. So basically, phenomic data is pretty much ready to be used in plant breeding program. And then this is another example, 0.5, when I use the multispectral data. So, and then this is the first results, and also and this is the one of the important part I would like to mention. I used 153,000 SMPs, and then I used the phenomic data and genomic data. If you look at just the CV4 part in here, genomic prediction predicted the untested genotype in untested environment with the accuracy of 0.55, and the other one is predicted pretty much same. So basically, performance of prediction is pretty much same. So I know I finished my time, but these are the variable importance scores we can extract. These are the area under curve for each vegetation indices, and we can use them in the GWAS to integrate with the GBS data. And this is the temporal genomic prediction, what is for each vegetation indices that are varying depending on the time point. And we published that in as a preprint if you are interested in the detail of this research. And also we predicted the senescence, what Aaron is going to mention today. And this is how we can visualize the senescence plot by using one of the late flight. And I am done, and thank you so much. And I can get your question if I have time, but I don't think that I have time right now.
Thank you, Alper. That was great. And it's good to know that we're going to slip right into what it is that Aaron talks about, and it relates. That will enable us after that last seminar to have a little bit of a discussion on what we see across the 2 seminars. How does that sound?
Of course, that's great. I will be in here.
Okay, thanks so much.
Of course.
Aaron DeSalvio is a PhD student also at Texas A&M. He's in an interdisciplinary graduate program in genetics. He received his bachelor's degree in May of 2021 after studying molecular and cellular biology at the University of Illinois at Urbana-Champaign. When not in class or at work, you can find him going for bike rides and practicing at the archery range. Nice introduction. You see Aaron's seminar title right there in front of you. And with that, I will turn it over to Aaron.
All right, thank you very much for the introduction. So the theme of this study is that we performed rust and senescence prediction in Maize using high throughput phenotyping data as well as machine learning algorithms. And so first we'll go over a southern rust outbreak that happened here in Texas this past summer, and then we'll look at senescence data from both 2020 and 2021. And then we'll go over the machine learning prediction models that we use to make our predictions. So to start out, southern rust is a biotrophic foliar pathogen that affects maize. And in the US, outbreaks have a history of causing up to 50% yield loss, and we've seen even higher rates than that overseas in past years. And so the main problem with scoring southern rust is it's very dependent on the person doing it, and it's time intensive, and it's only really scalable for research size plots. And so the image here shows some pictures of varying percentages of rust infection that I observed when scoring rust by hand in the field this summer. And so each score is an approximate percentage of the leaf area covered by rust. So rust can be really problematic if it's not identified early, and effective fungicide treatment is dependent on when it's identified. And so for future reference in this presentation, I should go over senescence, which is the deterioration of biological life, which we all more commonly know as aging. And in maize, senescence generally occurs after the completion of the reproductive phase. And so historically, the photosynthetic lifespan of leaves plays an important role in biomass accumulation. So stay green and premature senescence are important indicators that you can observe across various time points and plant growth stages. And in this study, we didn't take a traditional approach to scoring senescence. We wanted to try something new. And so instead of taking field level measurements, I placed a labeled grid over drone images from end of season maize and assigned scores between 0 and 5. And these were used as predictors for our machine learning models, which we'll discuss later. We had 2 scores from 2020 and 4 from 2021. And so these bottom 3 images are color manipulations that we performed to give you a better idea of which plants were senescing and which plants were showing stay green. And so it just shows that there's some simple tools in Photoshop you can use to accentuate otherwise, difficult to see phenotypic differences between plants. And before we dive into the rest of the presentation, here's just a quick overview of how we collected our data. So we flew a drone 13 times in 2020 and 17 times in 2021 at various days after planting. And this added a temporal component to our analysis. So when you think about the word temporal, just think about a profile of flights obtained across the growing season. And so I assigned senescence scores using mosaics for 2 dates in 2020 and for 4 dates in 2021. And rust was measured once on the ground in 2021. And so these are the dates that I performed those measurements. Okay, and next we'll go over a brief overview of our data analysis pipeline that we use from start to finish, which Alper kind of covered, but I'll just recap it here. So we, throughout the growing season, obtain images of the plants that are stitched together using GPS coordinate data. And this adds the temporal component to our analysis. So on top of these images, we overlay a field map using QGIS, and this defines the boundaries within which we extract our vegetation indices in RStudio. And we use the field image R package developed by our colleague Felipe Matias at North Dakota State. And so the next step involves taking these vegetation indices and building a random fit model in JUMP. And this allows us to obtain temporal best linear unbiased predictors, which, as Alper mentioned, we abbreviate as TBLUPs because it's a mouthful. And so TBLUPs are calculated using a nested model that nests each pedigree's performance within each flight. And so lastly, we take those TBLUPs along with, in this case, rust and senescence scores, and use those as predictors for our machine learning models. And so next we wanted to take a look at the contribution of each random effect in our model. And for context, the left axis is the explained percent variation by model component, and the right axis is temporal repeatability. And so the takeaway from this figure is that the green portion of each bar shows that the flight component obtained the highest percent experimental variation for all VIs in both years. And in this case, it ranged between 41 and 95%. And we expected that because we used the nested model design. And the black circles indicate the R-squared values, and the white diamonds indicate temporal repeatability. So after determining Pearson correlation coefficients for phenomic data between genotypes, we found that the correlation coefficients were strongest within the same growth stages, but weaker between growth stages. And this had an important implication. It told us that the temporal nature of the data we were collecting was more important than the vegetation indices themselves. And so this graph is the smooth conditional means for temporal breeding values of each pedigree for each vegetation index, with red being 2020 and blue being 2021. And the fact that we have so many time points to reference from our flights that we mentioned allows us to see patterns for plant developmental stages. And so next we're going to zoom in on two of these in particular. So the temporal breeding values tend to be most extreme at plant emergence, flowering times, and conclusion of the reproductive stages. And in these particular cases, you can see that some VIs follow a trend between years, as is shown down here, whereas others show variation between years. And this could be a consequence of the different temporal resolution that we had between the years, because we had 13 flights, 2020, 17, and 2021, but it's something we need to look into further. And so there are a few things to take note of in these images. And the left y-axis is the explained percent variation by component, and the right y-axis is repeatability, again shown by the white diamonds. Black circles again show R squared. And so the right side figure is set up the same way, but remember that since we scored senescence at multiple time points, there was a temporal component, so it's actually temporal repeatability. And rust had an approximately value of 0.9 for temporal repeatability, depending on the trial and the year, and senescence had temporal repeatability between 0.6 and 0.9 in both years. So something we didn't necessarily expect to find when we set out to do this study was a relationship between grain filling and yield. And so this we thought was kind of exciting. So we calculated grain filling by subtracting the date of flowering from senescence. And this revealed a correlation of up to 0.22 in 2020 and 0.44 in 2021. And these were the highest correlations between yield and than any of the other traits. And this signaled to us that grain filling may actually be a predictor of yield. And so this is something we want to look into in future studies. So this image is showing the temporal correlations between predicted variables and all vegetation indices. And so after looking at grain filling, we calculated the temporal correlations and of each vegetation index belonging to both years. And we found that if you look at particularly this figure on the right, the alternating colors reveal that early and mid growth stages had opposite temporal correlations for the four senescence dates in 2021. However, in 2020, the temporal correlations had more consistent trajectories. So that just shows variation between years. And now I wanted to clear up some terminology before we can continue, because there's a lot of words with predictive and predictors that kind of get confusing. So when we refer to a predictive model, we're talking about using predictors to train a machine learning algorithm. And so in this study, our predictors were the two senescence dates that we scored in 2020, the four that we scored in 2021, the hand measured rust score that I took in the field this summer in 2021, as well as the 36 vegetation indices we previously mentioned. And to be more precise, the T-BLUPs of those vegetation indices. And so we next partitioned the data set, as Alper mentioned, into 70% training data, which on the next slide you'll see this is labeled CV1, and 30% test data, which is labeled as CV2 on the next slide. And so the main difference is that when the model's in the training environment, it has access to genotypic information, but in the test environment, it loses access to that information. And that's how we can test the accuracy of the model. And so in this sense, the term prediction refers to what's happening within a data set. And so we ultimately like to get to a point someday where we can access a compendium of training data and make accurate predictions of entirely unknown genotypes. And I think that's not too far off in the future, but that's not what we did in this study. So looking at the model assessment that we just mentioned, this figure shows phenomic prediction accuracies for each model used to predict rust in this case. And just like the previous slide detailed, CV1 is the model's performance with training data, but the part to really pay attention here is CV2, which reveals the model's performance with test data in unknown genotypes, or untested genotypes rather. And so again, with test data, we're asking the model to predict the performance of those untested genotypes. And we can see that the linear model performed poorest in this case. And so on average, the models that performed best were elastic net, lasso, and random forest in our case. And so to me, what I'm going to present next is what I found to be the most exciting part of the study. And variable importance scores are a way of revealing how much the model depended on or used each variable to perform an accurate prediction. And so specifically, we're looking at which vegetation indices were most predictive of either senescence or rust, and the time and days after planting that they provided that predictive power. And so brighter red signifies that an index was more important for model accuracy. So in this case, we found that the red chromatic coordinate index was a critical vegetation index for both senescence and rust. And I'm just showing senescence here as an example. And in this case, you can see it acting as a phenotypic indicator as early as 17 days after planting. And so if we're able to validate this in future trials, this may enable both researchers and producers to identify rust infections before the classically recognized phenotypic signs emerge, which would allow for more flexibility with fungicide applications. And so some takeaways to leave you with from this study is that the work we did showed that traditional ground-based scoring may be a thing of the past. And so by using mosaic images, we're able to consistently score senescence in maize and use those scores as predictors for robust phenomic predictive models. And using the data captured at those scheduled intervals throughout the growing season, or our temporal data, in other words, machine learning models are able to figure out patterns that enable high prediction accuracy of the performance of untested genotypes for both rust and senescence. And by identifying robust indicators of disease progression in cereal crops, producers may be able to make more informed decisions about their management practices at a time. So some things to think about for the future. It's entirely possible that by using an RGB sensor, we may have missed out on a wavelength or multiple wavelengths of light, rather that better track disease and senescence progression in maize. And by performing concurrent multispectral and RGB flights, a similar study of this nature might do a better job at capturing senescence and rust progression. And as a result, there might be better vegetation indices that those would unlock that may serve as better predictors. So also in doing the study, we noticed that sensor settings may be playing a role in causing variation in quality between mosaics, between flight dates. And so by enacting stricter control over the sensor settings, or if any of you are photographers using manual mode, we might be able to obtain more consistency between mosaics and remove some of the unexplained model variance. And so there's also likely a point of diminishing returns when it comes to obtaining temporal data. So finding the sweet spot between image resolution and temporal resolution may ease the burden of data processing and storage for future studies. And lastly, there's undoubtedly other predictors that may supplement our existing models. And these could be, certain weather data metrics or soil composition data, and I'm sure there's others. And so the ultimate predictive model would incorporate all of those, as well as some of the predictors that we discussed today. And so I wanted to thank everyone involved in Dr. Murray's lab, especially Alper for being a great mentor this summer, and for everyone involved in managing the field. And thanks to all of you for listening. It's my first time doing this, so thanks for bearing with me. And I think maybe we'll have time for questions for both Alper and I's talks now.
Fantastic. Thank you, Aaron.
Thank you.
It's always a happy thing to me to have two seminars that we can have a discussion about, because they're so highly related. So for a first seminar, certainly nicely done, Aaron. Thank you again, Alper, for also very quickly deciding that you're able to give seminars this Friday. So it was just like a week ago that we actually threw this together. Lots of people are present. Thanks for that. Are there any questions here in this room or online? We have our chat up where everyone can see it. This is Colleen.
Hello, thank you guys for the great talks. I've got a question about the rust, and one of the images was where you hand scored them. How does flying over something where it's like 5% rust visible, is there like a sweet spot of a percentage for leaf coverage with rust that it picks up on that's still early enough where a treatment can be applied to it?
That's a great question. So I don't know the exact the exact biological phenomenon that's happening that allows it to be picked up that early. This is actually something I've been thinking a lot about lately, but we definitely need to look into more of like the molecular phenomena that are occurring there, if there are any. There was one study I found that particularly RCC was a good indicator for early detection of foliage in springtime for forestry, I guess, and they kind of combated the idea that it was just emergence of soil. There was so there's definitely some, I think there's definitely something at the molecular level happening. I'm not sure exactly what it is. I might have to learn more about the infection process of southern rust and what it's doing to the leaves. So I'm not sure yet, but we'd like to look at that in the future.
Great, thank you.
In our last series, this was a topic that came up for Adina Howe when she was looking at soil microorganisms and whether these microorganisms had anything to do with leaf phenotypes. So just as a comment, you may want to reach out to her. She may have thought about this some.
Thank you.
Sure. I see a note in our chat from Kat Holland.
Yeah, for rust.
Okay. Sorry, I got to pull that up.
And it looks like Seth has answered. So here's what the question was. For common rust of maize, younger plants are much more susceptible to infection. Is this the same for southern rust as well? That might help explain why that 17 days after pollination was found to have the high importance score as opposed to other time points. And Seth Murray has a comment of Kat, I don't think so. We have something, another question. So before we move on to the next question, are there any other comments on that one?
Well, I can tell something about like the answer for this question. First, statistic always gives a number for us. So when it comes to biological understanding, what we did in Aaron's study is pretty much new. We used the phenomic data, vegetation indices, temporal vegetation indices as predictors. And the variable importance score is what we can figure out which vegetation indices are most important belonging to which time point. So basically, all vegetation indices are related to how plant health are good. So basically, strong plant are kind of has a strong ability to deal with disease. But this year, we surprisingly find out very susceptible maize hybrid, which is northern adapted ones, but we didn't see these things in the southern adapted germplasm in here. So which is not surprising. So this is why we would like to fly the drones to predict the rust. So we need to dissect this situation better. But yes, there are some early indicators. 17 days after planting RCC scores is really interesting, for instance. This is how I can answer this question.
Thank you, Alper.
Of course, there's another question about field image R, and the question is perfect, by the way. Weeds problem is, I spent like last 3 and 4 years to have a smooth pipeline. There is no smooth pipeline from my point of view. Weed is always a problem. So yes, there is a couple of function where you can smooth the bad effect of weed, but you cannot remove the weed effect entirely. So the best way is hoeing. I can say that.
So Alper is answering a question that was online that has to do with whether the presence of weeds is a problem for field imager. Is that right?
Yes, this is the answer for this question. The tonic, if I'm not pronouncing wrong, and Dr. Boris, okay, he also helped us.
So the next question from Felix to everyone says, I was wondering, when it's about predicting new genotypes, can you actually say it's like this? Because you can only test for your own genotype data set. So how accurate is this prediction in the first place? How much does genotype variation, how much variation is there?
Okay, I understood. So basically, this question is referring to what genomic prediction or selection can do. So I recently finished a manuscript about that. What we did in there is, for instance, think about that you train a model by using 100 set of Maize hybrid in this year by using the Phenomic data, and you would like to predict totally unrelated and unknown phenotype data in 5 years later, and then but the flight time are not matching, and these are problems. So now we try to figure that out, how we can relate 2 different years to predict, to realize the whatever the question says to us. And basically we did that. So for instance, there are a couple of ways we can do. We can use the principal component scores. We can use the spline method to fit the temporal data, and we can match 2 different years to predict totally unrelated and the unknown genotype. Yes, we did, and the accuracy is around 0.4 or 0.5, and there are some hopeful results in there. But we need to create a flow chart to show how phenomic prediction can be applied to real plant breeding program. We are about to do that, but I mean, right now, this is how I can answer for this question.
Felix, does that answer your question? Fantastic. Okay. So let's go back up. We got through that. Question for Aaron or Alper. Why you planned to compare or use multiple vegetation indices in your studies? My next question, as there are hundreds of 6 VI in market nowadays. So on what basis do you select those vegetative indices for your study?
I can go first, Aaron, if you want.
Okay.
You can prepare your own answer for sure. So yes, we created all these vegetation indices by using either 3 or 5 different beds. This is what we can do best first to create predictors. But when it comes to machine learning prediction, somehow the beta term, especially if you're interested in machine learning, beta term never goes drop in rich regression or in loss. So beta term sometimes can be dropped towards the 0 to drop any unnecessary predictors. And Aaron showed a prediction, the correlation of the temporal vegetation indices. Yes, there are some vegetation indices are correlating between one another, some are not. So yes, there are a bunch of vegetation indices in the market nowadays, and we can try that. But the important is that multicollinearity problem. Is there a multicollinearity problem or not? So in order to assess that, simple correlation can answer this question. But when it comes to genomic prediction, we are creating our diversity based on biparental population or magic population or NAM population. We are using basically limited number of parental lines. So the situation, there are same situation in there. So, but I mean, in general, there are multicollinearity in phenomic and the genomic data. But I mean, I just try to compare genomic prediction accuracy and phenomic prediction accuracy, and they are comparable. This is the first step, but we need to evaluate which vegetation indices are more important. But more than that, temporal, time points are more important than vegetation indices. This is what I wanted to underline in my presentation as well.
Thank you. Are there questions I'll ask first in the room that have popped into your heads while we've been going back and forth with online comments and questions? Okay, how about for those that are online? Do you have any remaining burning questions? If not, let's thank Alper and Aaron for presenting for us today. I hope you could hear that clapping in the room.
Of course.
It really happened.
Yes, thank
I
you.
heard clearly. Thank you so much.
Thank you for joining us. Thank you for preparing such great presentations. And I especially like how those two presentations were in the same space. I think it helps for situations like this where we're only together for a short period of time.
Of course.
Thanks.
Thank you for having us.
Thank you so much.
We look forward to seeing many of you and a few others if you'll encourage others to join us next Friday, same bat time, same bat place. Bye-bye.